Hi everyone, my name's Andre. Welcome to Med School EU. And the topic of today's video is going to be the nerve pathway and action potentials of the nervous system. So today we're going to talk about the second part of the nervous system unit, and we're going to discuss the connection between neurons, which is called the synapse, and we're going to talk about action potentials. And overall, we're just going to discuss how signals are actually transmitted along the nervous system. So first of all, we're going to discuss the nerve pathways, how nerves are structured in our autonomic nervous system, so the one that is involuntarily controlled and is completely controlled by our brain. So the signals that are being sent out by the brain are not conscious signals. We're not aware of them, and this happens automatically. We don't need to think about it. So the way our organs are activated based on their activity in order to increase their activity or decrease their activity is activated by the autonomic nervous system, which would stem to be the parasympathetic and the sympathetic division. First of all, this is called the nerve pathway. We are going to discuss certain details about this because this actually came up on the 2021 IMAT exam. You actually had to know some of this information in order to answer one of the questions. So this the one that comes off the spinal cord or the central nervous system is called the preganglionic neuron. So preganglionic, and it's going to make more sense why it's called preganglionic, because this little bundle right here is called the autonomic ganglion, and this preganglionic fiber is myelinated, and we talked in our previous lecture about the myelin sheath that is composed of Schwann cells. So the Schwann cells, basically, they provide insulation for these fibers, these nerves, so they are able to transmit their signal much faster. So the preganglionic fibers are myelinated, meaning that they are going to have these Schwann cells wrapped around them. As you can see here, the Schwann cell, Schwann cell, Schwann cell, and they're going to have nodes of Ranvier going right in between them, so the signal can just jump between one to the other and transmit much faster. Now the postganglionic fiber does not have the myelinated sheath. It is unmyelinated. Also, the acetylcholine right here and the norepinephrine, these are neurotransmitters that are used to transmit the signal. These are chemical signals that are going to be able to be absorbed by the organ, and then the activation of further action potential. And here at the autonomic ganglion, the signal will be transmitted only by the acetylcholine, not norepinephrine. So now, looking at both sides, we've got the sympathetic on this side on the left, and the parasympathetic, so P and S, and we got S and S. Sympathetic nervous system, parasympathetic nervous system. We discussed these in great detail in our last lecture. So the sympathetic nervous system is going to come off from the thoracic vertebrae, from the spinal cord. T1 to T12 stands for the thoracic vertebrae. And these nerves, these pathways are going to come out of there, and they're going to synapse at the sympathetic chain of ganglia. So this is called sympathetic chain of ganglia that will be located close to the start of the preganglionic fiber. So as you can see, the preganglionic neurons are going to be quite short compared to the parasympathetic nervous system, because these are going to go directly to the organs or possibly go very close to the organs we're going to synapse. However, in sympathetic nervous system, these sympathetic chain ganglia are going to be much closer, which will make the preganglionic fibers much shorter. So they're going to be short, but they will have long postganglionic fibers. Now these ones are called collateral ganglia, where the preganglionic fibers are going to go past the sympathetic chain of ganglia, and they're going to synapse at collateral ganglia. Now, as you can see, the preganglionic fibers are never going to reach any of the organs on their own. They're going to have to synapse at some sort of ganglia in order to reach the final destination. However, the parasympathetic nervous system is going to be coming off the brainstem. So these are actually going to be the cranial nerve. So this would be cranial nerve number 3, cranial nerve number 10, which is the vagus nerve, very important for our body. And they will also be coming from the pelvis as well. So that's the two locations where the parasympathetic nervous system is coming from. That's the pelvis and the brainstem, whereas sympathetic are coming off from the thoracic vertebrae. Now, what's important to know is that the parasympathetic nervous system has long preganglionic fibers that will likely not be attaching to any ganglia. However, the sympathetic will have long postganglionic fibers in comparison. As you can see, the postganglionic fibers here from the parasympathetic nervous system are extremely short, if at all, because over here they will be bonded very close to the organ. So that's important to note. That's the distinction that you needed to know to answer one of the questions on the 2021 IMAT exam. So I would suggest you memorize this diagram and you memorize these concepts by heart so you can avoid mistakes on this year's exam. So we are going to discuss a little bit about the resting action potential. So basically we are going to have a membrane. This is the membrane. And we are going to have our pumps. So this is going to be, this blue one right here is going to be the pump that pumps ions in and out of the membrane, which keeps the membrane either positive or negatively charged. And we're going to discover what actually happens here. So what we will have is the sodium will be pumped out of the cell. So if we're going to label this as the extracellular fluid and this is the cytoplasm, which would mean that this is the inside of the cell, the sodium Na+ will be pumped out of the cell into the extracellular fluid, and the potassium from outside the cell is going to be brought into the cell. So, and it's going to be done at the rate of 3 to 2. So 3 sodium will be pumped out of the cell and 2 potassium will be pumped into the cell. And this is the mechanism that will be used, the sodium-potassium pump protein that will be used in order to keep the resting potential at -70 millivolts. So the membrane potential of before any action begins, before any electrical impulse could be transmitted across a cell, they are going to have to be kept at this -70 millivolts because more, as you can see, 3 sodium, more positive charges are flowing out of the cell than they are coming into the cell. So it's going to maintain this sort of electrochemical gradient. In addition, this gradient of -70 millivolts will be maintained by the potassium leaving or leaking through the membrane at a much faster rate than the sodium coming back in, because the sodium concentration inside the cell is low and the sodium concentration outside the cell is high. So this action pump, this sodium-potassium pump protein, is active transport because it's pumping against its chemical gradient. So it's going against the electrochemical gradient, and same with the potassium here, because the potassium is coming in to a high concentration from a low concentration on the outside. So it's naturally going to want to diffuse out into the extracellular fluid. And because the membrane is leaky, meaning that it leaks our potassium slowly and at a certain pace, it leaks more positive charges but it's not as permeable to sodium for having it leak back inside the cell. So therefore it kind of maintains, this whole structure works on maintaining this -70 resting active potential. So generally the cell is more negative on the inside than it is on the outside, and this will be the typical driving force of the action potential, why it even is a phenomenon. So now we are going to talk a little bit about the action potential. So we know that our membrane potential is -70 millivolts, and if we take a look at a chart or a graph of this and try to kind of depict what really is going on in terms of the potential difference. So here we're going to have millivolts on this side, and here the time frame. And we would begin with the -70 because that's where membrane potential is. And here's the key with the +30. So typically the cell nicely resides in this -70. However, when a stimulus occurs, when electrical stimulus comes through the cell onto its cell membrane, it's going to cause an influx of positive energy, which is going to grow. And then once it crosses this -50 threshold, it is going to cause an action potential. But it has to reach this threshold, because if the action potential is not strong enough and it reaches just underneath it, it does not reach this threshold, an action potential is not going to occur. So these little stimuli are not going to cause an action potential. They're not going to cause the cell to do anything about its environment. However, if we want the cell to activate through the electrical signal and pass on the stimulus, it's going to go above the 50 mark, and then it's going to cause an influx of positive energy, which will then cause a slow decline and an overshoot, meaning it's going to go below -70, and then it will realign itself back to its original membrane potential. Now the key here is that it's going to revert from -70 millivolts to +30. So it's going to be influx of positive energy. Now all of this is caused by the changes in the permeability of the membrane, and that is going to cause the sodium ions and the potassium ions to be going across the cell membrane at a very fast pace. So when the cell is at its resting potential, these pumps and these channels between, so these sodium and potassium channels that are associated in the membrane, they are going to be closed. So they're closed when the membrane potential is -70. They're closed off. They're not going to allow the sodium and potassium ions to leak quickly through from one side to another. The potential is going to be maintained again by the sodium potassium pump, and these channels are going to be completely closed. So what happens during an action potential? First, the electric current used to stimulate the axon causes the opening of the voltage-gated channels in the cell surface membrane, which is going to allow this sodium ions to pass through. So we get an influx of sodium ions. Now obviously sodium ions are going to make the membrane or the inside of the cell, the cytoplasm, more positive. So it's going to go from -70 to more positive side, -50, -30, and so on. And because there's a much greater concentration of sodium ions outside the axon than on the inside, the sodium ions enter through these open channels that were previously closed. Now they're open because they were stimulated by voltage. Remember, these are voltage-gated channels, and so therefore the sodium potassium pumps are not going to be as effective because these channels are going to be open and they're going to be leaking ions all over the place. So the sodium ions will leak through from the outside to the inside of the cell, causing the cell to become more positive compared to its environment. And this part is called depolarization. So this aspect right here is called depolarization. repolarization, and it is done by the sodium influx inside the cell. And so there's this threshold, as I've mentioned before. If this threshold is passed, so the depolarization originally happens a little bit slower, but as soon as it is passed, it causes a huge influx because more and more are becoming open. And if the potential difference reaches about negative 50 millivolts, then all of these channels are going to open, and the potential will then quickly appear to be plus 30 millivolts compared to the outside. So after about 1 millisecond, so after this would be about 1 millisecond where this action potential becomes plus 30 millivolts, that's the potential difference between the inside of the cell and the outside, the sodium ion voltage gated channels close. So the sodium ion stop diffusing into the axon, and at the same time the potassium ion channels open. So at this point the potassium ion channels are closed, meaning that the positive charge that's already inside the cell is not going to be leaking out to the outside. Only the sodium will be leaking into the cell, as you can see right here by this. And that's part of depolarization, and that's what we highlighted here. However, once it reaches the plus 30 and the action potential has already occurred, what we're going to get is something called repolarization. And during repolarization, we are going to have the sodium ion voltage gated channels are going to be closed, so less sodium will be coming into the cell and more potassium will come out of the cell. So the positive charges will be leaving the cell and less positive charges will be coming into the cell, because now the potassium ion channels are open and the sodium channels are closed. So as you can imagine here, we're going to have an influx of positive charge that's going to be coming out of the cell. It's going to be leaving the cell, meaning that the cell is going to become more and more, progressively more and more negative, because it removes the positive charge from inside of the axon to the outside. Therefore, it's going to return the potential difference to minus 70, and that's the whole process of repolarization. However, the potential difference across the cell briefly causes an influx of this positive charge leaking out, and therefore it's going to cause a hypershoot, which is going to be called hyperpolarization, because the positive charge will be leaking out at such a fast pace that it's going to cause an overshoot, and as you can see, it's going to go below minus 70 millivolts. So again, a couple of concepts you need to remember. I'm going to summarize this because this is something that's extremely important for the test, and it's something that you should know by heart. So in order for an action potential to occur, the axon membrane potential must be depolarized. They must go from negative 70 to plus 30. However, in order for that to happen, the stimulus, the electrical stimulus, must be strong enough that it reaches above negative 50 millivolts. Once it reaches above negative 50, it is past the threshold. So this is the threshold potential, and once it's passed, then the sodium will leak in more and more, and more positive charge will be inside of the axon. And this all occurs, this depolarization occurs, why? Because the sodium ion voltage gated channels are open while the potassium voltage gated channels are closed. However, once it reaches this 1 millisecond time and it has caused this action potential, the sodium channels are going to close and potassium channels are now going to open, meaning that the positive charge will begin leaking out, because potassium has the chemical difference, electrochemical gradient, going from the inside of cell towards the outside. So it wants to leave because that's where there's less potassium. So it's going to go from higher concentration to lower concentration by just general diffusion. So potassium is going to leave the cell, the axon, because these channels are now open. It allows them to just leave. And the sodium will then not be able to leak into the cell from the outside, since the outside is more concentrated than the inside, and therefore you're going to get less positive coming in and more positive leaving the cell, and this part is called repolarization. Now I wanted to revisit this concept again to understand why depolarization and repolarization really occurs. So if we have our membrane here, we have the cytoplasm and we have our extracellular fluid, what we have is this enzyme, which is just going to be a protein. It's going to act as a pump of sodium and potassium. There is a higher concentration of sodium outside of the cell. Keep that in mind. And there's a lower concentration of sodium on the inside of the cell. Now, because this pump is an active pump, it's active transport, it's going to go against its electrochemical gradient, and it's going to pump 3 sodium outside the cell even though there's already low amount of sodium on the inside of the cell. Now potassium is the opposite. We got low concentration of potassium on the outside and high concentration of potassium on the inside, and the way it's going to work is, again, it's going to pump against its concentration gradient and bring the positive inside. Now what happens during depolarization? During depolarization, we're going to get these other channels. They're called voltage-gated channels because they respond to voltages. They respond to electrical stimuli. And these channels are going to open. The sodium channels will open once electrical stimulus goes through, and they're going to open, and the sodium is now going to go down its concentration gradient from higher concentration down to the lower concentration, which is inside the cell. So it's going to cause the cell to be more positive. So from -70, it's going to begin to rise towards -50, and then once it passes that threshold, it's going to go to +30 millivolts. Because this sodium ion voltage gated channel is open. However, once we reach the peak at +30, we're going to get repolarization, and during repolarization the opposite occurs. Sodium ion voltage gated channels are going to close. So right here they're just going to close. These will be closed, and these channels are going to open. So the potassium channels are going to now open, and the potassium channels will force the potassium to go down its concentration gradient, going from the inside of the cell to the outside of the cell. So more and more potassium, more positive charge will be leaving, and then we are going to have our membrane potential go right back to -70 and do an overshoot, which is going to be hyperpolarization below -70 because of this crazy outflow of positive charge. And no positive charge is coming in. The sodium's not coming in because these are inhibited, these are blocked, and so therefore the potassium's going to leave. And that's how an action potential occurs. This is how it occurs down the axon, where it goes from one node of Renvier to the next node of Renvier. And that's how the membrane is going to be depolarized and how it's going to activate the signal, and it's going to be transmitted from one part of the cell to the next part. Now I also wanted to address several misconceptions about action potentials, because I believe it's extremely important in the way that you're going to understand what an action potential is and how a signal is actually transmitted and how it carries information, how we're able to perceive that something is bright light versus dim light, or something is a strong stimuli versus a weak stimuli. And it's not going to be perceived by the action potential size. So the size of the action potential is not going to change. It will remain constant. It is not going to be changing throughout the axon. Nor do they change in size according to the intensity of the stimulus. So intensity is another variable that is also going to be constant. The action potential will continue to reach the peak value of plus 30 millivolts inside all the way along the axon. So if it's traveling from the spinal cord all the way down to the toe, it is continuously going to produce plus 30 millivolts throughout the entire length of the axon. Now, if you have a very strong light shining in your eyes, it's going to produce action potentials of precisely the same size as dim light. So again, size is not going to be a variable because the size is always going to be the same in terms of the action potential. Even if it's bright light or it's dim light, it's still going to have the same stimuli. Or if it's hot stove or if it's cold stove, you're going to perceive the same size of action potentials. They're not going to be the difference that distinguishes these two stimuli. And the speed at which action potentials travel does not vary according to the size of the stimulus. Well, what is different about action potentials resulting from strong and a weak stimulus is the frequency. So the frequency of stimuli is the most important thing that distinguishes between a strong stimuli and a weak stimuli. So something like bright light will have the frequency of like this. It's going to have stimuli, 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 whereas the dim light will have a frequency of 1, 1, 1, 1. It will come at a lower frequency. It will not be as often stimulating the nerve. But the nerve, it will be stimulated very often in terms of the action potential. So it's going to be creating new action potentials over and over and over again at a greater frequency if the stimulus is strong. So bright light versus dim light. Now another thing we must talk about is the speed of transmission. Now the speed of transmission I already mentioned. It depends on the myelination, so myelination of the axons. So if the axon is unmyelinated, it's going to travel much slower than in a myelinated axon. So if they have these nodes of Renvier, the stimuli is going to travel way faster. Another thing is going to be diameter of the axon. Diameter of the axon. So of course, the larger the axon, let's say it's this wide, it's going to have less of constriction. It's going to be more dilated, so it's going to be bigger. It's going to have a bigger volume to fit, and therefore it's going to have lower resistance. So the stimuli will just travel freely. However, if it is a tiny neuron or the tiny axon, it's going to be hard for the stimuli to push through, so it's going to have a higher resistance in this case. So the diameter of the axon is also going to be of high importance. Now in terms of myelination, the way that the signal is being conducted, so it's going to be going between these nodes of Renvier, which are the nodes that are in between these myelinations, and this is going to be called saltatory conduction. In a myelinated axon, saltatory conduction can increase the speed of transmission of up to 50 times than in an unmyelinated axon of the same diameter. So it's very important that our nervous system has these myelinations from the Schwann cells, because they are able to provide much faster signals, and the transmission, the speed of transmission is going to be way, way faster. Now the final concept that we're going to discuss today is going to be a synapse. And what's a synapse? It is basically the connection between one neuron and another neuron. So the terminal ends and the dendrites are going to be connected to form a synapse. So we're going to zoom in here and see what's going on and how a synapse really occurs. So first, we're going to have this action potential that we just talked about. The action potential is going to reach the end of the axon at the axon terminal and depolarize its membrane. So the action potentials will be going down this way, and it's going to depolarize this membrane. Now, as soon as the membrane is going to be depolarized, the voltage gated, remember the voltage gated are the ones that will open when a voltage passes through, so an action potential passes through. We have an action potential. It arrives and it's going to open the voltage gated calcium channels. Now the calcium from the outside, from the synaptic cleft, is going to flow inside over here. It's going to flow inside to the axon terminal. Now the calcium influx triggers synaptic vesicles to release neurotransmitters. So these circles right here, they're called synaptic vesicles, and these vesicles are going to be holding neurotransmitter. And I've mentioned about neurotransmitters previously in our last lecture, how the preganglionic fibers are going to attach to the ganglion and they're going to release acetylcholine. However, the postganglionic fibers are going to release acetylcholine or norepinephrine. Now these are going to be typically acetylcholine, so these are neurotransmitters. And what's going to happen is these neurotransmitters are now going to fuse with the membrane. Right here, they're going to fuse with the membrane, they're going to do exocytosis, and now there's going to be plenty of these neurotransmitters. And the neurotransmitter binds to receptors, so it's going to bind right here to these receptors on the target cell, and in this case it's going to cause positive ions to flow in. So we're going to have positive ions, plus here, plus and plus and plus, and all of that is going to flow into the cell. Now of course, this influx of positive energy is going to cause depolarization, and it's more likely going to cause an action potential. And by the way, these are not voltage gated channels, and their actual name is going to be ligand gated channels, and these channels are not going to be exposed to voltage, so they're not going to be gated by voltage. They're only going to be influenced by a neurotransmitter, which is going to be acetylcholine here. So the white parts that are released here are acetylcholine, and what it's going to do is it will cause the ligand gated channels to open and cause an influx of sodium ions, which will cause another action potential on the receiving neuron. Now something very similar is going to be happening with the muscle stimulation, and this is something that we're going to talk about in our next video.